All right, it's just 2 minutes past. Allow me to welcome everybody to the webinar. We welcome you. This is the second webinar for the data science series. Thank you so much for making time to join us for this series. We thank you so much for the joint organization, and we send our sincere gratitude to our speaker today, who is Dr. Oliver Payne. A brief introduction about this series. They are jointly developed by the Sickle Cell Disease Data Coordinating Center, which is SADC, and it is housed at the University of Cape Town in South Africa. SADC is generously funded by the National Heart, Lung and Blood Institute of the US National Institutes of Health. It's also jointly developed by the Human Health and Heredity in Africa Consortium, that's H3 Africa, as well as the African Society of Human Genetics and Wellcome Connect Science. Well, the aim of this webinar series is to help highlight the ongoing research in data science and biomedical research, and it's aimed to expose all of us who are attending to cut-edging methods and approaches in data science for biomedical and epidemiological research for our continent. So this series is also leading up to the 15th African Society of Human Genetics and the first Ugandan Society of Human Genetics and Bioinformatics. A link is going to be shared for those who have not yet sent in their abstracts or applied for the travel fellowships. The deadline is 15th November, and that's this week on Friday. Please send in your work. We'll be happy to come together to discuss cut-edging research on the continent.
How are you?
So a brief introduction about our speaker today. Dr. Oliver Payne completed his bachelor's in molecular genetics, followed by a master's in genes, environment, and development. He was awarded his PhD in 2017 from Birkbeck College, specializing in the genetic epidemiology of adolescent psychotic experiences. Dr. Oliver's postdoctoral journey included positions at Cardiff University and King's College London, leading to his current role as a Sir Henry Wellcome postdoctoral research fellow at the Morris Hall Clinical Neuroscience Institute, and this is since 2021. So Oliver specializes in statistical genetics, focusing on Genome Wide Association Studies to uncover the mechanisms underlying psychiatric and neurological conditions, and these include autism, antidepressant response, and motor neuron disease. His research integrates functional genomic annotations to advance understanding of disease etiology, to discover new therapeutic targets, and improve genetic prediction of clinical outcomes. A very significant contribution of Oliver's work is the development of the publicly available software pipelines, such as the GenoPred that he's going to be talking about today. A link to is going to be shared about this pipeline. So throughout this webinar, we encourage everyone to utilize the chat box in case you have any questions to ask. And please keep yourselves muted at all times so that we do not interrupt Oliver as he presents. Thank you so much. Allow me welcome Oliver to share his screen and start the presentation.
Wonderful. Thank you very much for the kind introduction, Gloria. Just loading up my screen now. Can you see my slides?
Yes, we can see your slides.
Wonderful. So, hello everyone. It's an absolute pleasure to be presenting here today. So as was hinted, I'm going to be talking about kind of GWAS, Genome Wide Association Studies, polygenic scores, and then highlight some of the work I've been doing regarding the development of a software pipeline called GenoPred, which is a pipeline for calculating polygenic scores. So I'm going to start by my slides.
Doc, please, we can't hear you.
You can't hear me. Okay. Did you say you can't hear me? Sounds good, no problem. Okay, so I think it might be a bit of a issue on that person's end.
Dr. Oliver, we can hear you well. You can go ahead.
Okay, wonderful. I'm trying to get out of this presenter view now. Okay, so the structure of the talk today is I'm going to give a bit of an introduction to polygenic scoring, first of all, because I know there's a range of backgrounds listening today. Then talk about GenoPred, this pipeline I've developed, and talk about some of the next steps for that work. So I'll start by, at the beginning, which is there's substantial evidence that genetic variation leads to a wide array of individual differences in health and disease. And one approach for identifying the specific genetic variants associated with the outcome of interest is referred to as a GWAS, which stands for a Genome Wide Association Study. And the way these GWAS work is by collecting large groups of individuals, then measuring the outcome of interest. For example, you could look at height, and you also assess them for genetic variants across the genome. And these genetic variants are usually single nucleotide polymorphisms where a single kind of letter in the DNA varies in the population. And then you test for an association between that outcome of interest and each genetic variant across the genome, hence why it's called a Genome Wide Association Study. And what you get at the end of that is what's referred to as GWAS summary statistics. So these are essentially association results for each variant and your outcome of interest. This is an example of what they could look like. So you usually have some information on the location of the genetic variant. So the chromosome number, an ID, base pair position. You then have information on which kind of allele or version of the genetic variant you are testing the effect of. And then you'll have an effect size, which could be a beta or an odds ratio if you're looking at a binary outcome, some kind of error estimate, sample size, p-value. So this is just a little animation from the GWAS catalog, which is a website storing genome-wide significant associations published in the literature. And each of these white lines of chromosomes and these little colored dots coming off them are significant genetic associations. And this is just showing that over the last 10, 15 years, 20 years, there's been a huge explosion in the number of robust genetic associations identified for a very broad range of outcomes. And this figure is already well out of date, and you can't even plot it anymore. There's a huge wealth of information available. And all of this, or the vast majority of it, is publicly available data. So, what can we do with all of this incredible information we have on how genetic variants are associated with health-related outcomes? Well, there's lots of things you can do. But one of them is called polygenic scoring. So a polygenic score summarizes an individual's genetic risk for a given outcome. And it's typically in its simplest form calculated as the sum of the sum of risk variance carried by an individual weighted by the GWAS effect size. So there's a very basic schematic of this on the right-hand side. Here we have our GWAS summary statistics with an effect size for each variant. And we could look at an individual's genetic information and just add up the number of these variants the individual carries, weighting those variants by their effect size. And that then gives you a polygenic score for that individual, which you can compare to the general population to determine where they lie in the kind of distribution of genetic risk for the traits that you are interested in. Now, there's a huge range of applications for polygenic scores in both research, but also personalized medicine. Here's some examples of that. So in personalized medicine, you could calculate a polygenic score capturing an individual's risk of developing a given disease in the future, which could be very useful for putting in place early interventions to avoid that risk or reduce that risk. You could also potentially gain insight into the prognosis of an individual's disease by predicting things like how long are they likely to survive, or what are the likely what's the progression of the symptoms going to be. And also you could also look at treatment. So there's now GWAS studies looking at response to medications, and you can use polygenic scores to predict whether an individual is likely to respond to a given treatment, which is all really valuable information when trying to give information to a patient or someone coming into the doctor's office. I'll just say now, of course, genetics doesn't explain everything. In fact, for some things, it's very difficult to use genetics to predict them. So I'm not saying that polygenic scores are going to be the thing that can predict everything. It's really just a tool for enhancing our ability to personalize medicine. So it'll be in addition to other clinical variables that we're already using to try and give individuals the right advice. There's also a range of things you can do with polygenic scores from a research perspective. One of the main ones that people use it for is looking for evidence of genetic overlap between outcomes. And I give an example of this later on. There's also other things you can do with them, such as controlling for genetic effects. So you could imagine in a clinical trial, perhaps, you would want to remove the effect of genetics to see more clearly what the effect is of the intervention. So that's an example. So there's lots of things you can do with polygenic scores. I'm going to give some examples of that now. So I mentioned you can predict risk of disease. This is a good paper to cite when you're trying to make this point, because they really nicely show that polygenic scores can be used to identify quite large groups of the population at significantly elevated risk of a range of common diseases. In the figure on the right, this is just showing the results for coronary artery disease. But essentially what it's saying is that the polygenic score for coronary artery disease on the x-axis, if you look at individuals in the top 8% of that polygenic score distribution, those individuals above that threshold have a 3-fold increase in their risk of developing coronary artery disease. So that's a very substantial increase in risk for quite a large proportion of individuals just identified using a polygenic score. So it kind of showcases how polygenic scores can be used to identify individuals that are at high risk for common diseases. This is a really nice paper as well, also looking at coronary artery disease, which shows that polygenic scores can be used to predict the efficacy of a drug or response to that drug. So they're looking at coronary artery disease, and they're using clinical trial data, looking at the effect of statins versus the placebo. And they split their cohort into individuals with low risk and high genetic risk based on a polygenic score. And there's two things you can see here. One is that on the y-axis, we have the event rate, the disease event rate, so something like a heart attack. And what you can see here is that, first of all, individuals in the high genetic risk plot are on average more likely to have a disease event regardless of which drug they're given, which makes sense because they have high genetic risk for coronary artery disease. But also, when you look at the effect size of the statins, if you compare the effect of being given a statin to the placebo, the decrease in risk is larger in the high genetic risk group, indicating that individuals with high genetic risk, although they have a higher probability of having the disease event, they're also going to have a greater response to the medication. So this is quite a nice example of that. And building on the kind of coronary artery disease literature a bit further, there's been very recent research in the UK showing that these polygenic scores can be used to improve prediction over and above existing clinical risk calculators. So for coronary artery disease, the one that's currently used is the QRisk-2, and they've shown that polygenic scores for coronary artery disease, when integrated into the kind of QRisk model, improves prediction of risk, but also importantly, it was well accepted by the kind of clinical, by the doctors that were giving that advice, and also by the patients in understanding that advice. So all in all, this is the first step, a really important step into actually starting to use these polygenic scores in the clinical setting. One final example on applications of polygenic scores is looking at genetic overlap between outcomes or genetic correlation. So this is quite a fun study. There's many more serious studies using it than for this purpose, but it's just an interesting one to look at. And what they're showing here is on the y-axis, you have the variance explained by the polygenic score. And on the x-axis, you're using a range of kind of GWAS thresholds to select the variance included in the polygenic score. But essentially what you see here is that the variance explained by the schizophrenia polygenic score can predict schizophrenia. This is a plot for bipolar disorder, showing that the bipolar disorder polygenic score can predict bipolar disorder. But then they also use these schizophrenia and bipolar disorder polygenic scores to predict creativity. So, you know, I don't know how they measured that even, but probably artistic traits or something like that. And they show that actually these genetic risk scores for psychotic disorders can predict creativity, indicating that there's a genetic overlap between being creative and a risk of schizophrenia. So a bit you know, bit of a weird one to interpret, but hopefully that's a nice demonstration of it. So those are the applications of polygenic scores. There's probably many more than that. And I just want to give a little insight into some of the more complex methodological considerations of actually calculating these polygenic scores. So one thing is that genetic variants that are nearby one each other in the genome are in LD, which stands for linkage disequilibrium. And it's essentially saying that there's a correlation between nearby variants. So if you have two genetic variants that are very close to each other, chances are they'll kind of co-occur. When you look at an individual, if they have one, they'll have the other. And it's very important to account for this linkage disequilibrium when you're calculating a polygenic score. And this is illustrated in the figure on the top right here. So what we're seeing here is a kind of very zoomed in version of GWAS results. So we're looking at one small region of the genome. On the y-axis, we're seeing the minus log 10 p-value of the variance in that region. So points that are high on the scale are more associated with the outcome of interest, so the GWAS outcome. And the color of the points indicates the correlation between the variants in the region and the index variant, which is this variant at the very top here. And what you can see is that there's this kind of peak of genetic associations, but there's a strong correlation between those genetic variants, so they are not independent associations. So if you don't account for the LD, you're going to end up double counting genetic effects in areas where there's this correlation. So it's very important to either remove variants that are in LD and just retain the best variant, or you can estimate the effect of these variants jointly. So kind of model their joint effect, thereby accounting for the correlation between them. So that's one complexity of polygenic scoring. The other is you need to consider the genetic architecture of the outcome. And genetic architecture describes the distribution of genetic effects. So for some diseases, there's just one variant that has a very large effect on the risk of that disease. For example, Huntington's disease. That's like a classic monogenic disease. Of course, it's more complex than that, but there is a very large effect of one locus in particular. So that's not, that's one end of the scale. And then you have other things like height, which there's no big effects. There's actually just many, many, many very tiny genetic effects. And this is referred to as so height would be therefore referred to as a highly polygenic trait. So this difference in kind of distribution of genetic effects, or this difference in polygenicity, needs to be taken into consideration when deriving the polygenic score, because there's a lot of noise in a GWAS. You can see in this figure on the top right, again, there's lots of genetic variants that are associated to some extent, but we don't have much confidence in them because the p-value is quite large. So usually people apply some sort of filter or some sort of shrinkage to get rid of variants that are kind of no consequence on the trait. And that where you place that filter or the strength of the shrinkage will depend on the polygenicity of the outcome. So yeah, those are the two kind of important considerations. This bottom right figure just illustrates those two scenarios of either being monogenic or highly polygenic, and it's the change in architecture is usually due to the amount of negative selection on the trait. So throughout evolution, some traits are selected for and against, and that will impact the kind of presence of large genetic effects, and vice versa. So another quirk of polygenic scoring is that we need to do all of this with summary statistics only. So we can't just take individual level data with all those genetic variants measured and all the phenotype, all the outcomes measured, because genetic data is private data and it cannot be shared easily. But we also need very large sample sizes to find robust associations. So what the whole field has resorted to is using these summary statistics, which are not individual level and can therefore be shared very easily. But this means we often need to use these summary statistics in combination with other reference information capturing the linkage to equilibrium kind of patterns to derive our polygenic scores. So that's kind of a fun quirk of polygenic scoring. Like all other kind of machine learning or prediction modeling fields typically use individual level data, whereas in the GWAS world, we're only using summary statistics. So one thing I haven't mentioned yet, which is very important, is that there's a huge bias in the GWAS literature towards individuals of European ancestry. So the vast majority of people that have been included in GWAS are from European populations. And if you try and apply results from one population into another through polygenic scoring, you'll see that polygenic scores will perform worse in populations that are different to that of the GWAS. So for example, in this plot, they derive polygenic scores using European GWAS summary statistics, and they're showing the relative performance of polygenic scores in Europeans, Central Americans, South Asians, East Asians, and Africans. And you can see that as you move further away in a kind of ancestry-related way from Europe, the prediction from the polygenic scores gets worse. And this needs to be kind of addressed, because if we start using these polygenic scores in a clinical setting, then there's going to be an exacerbation of health inequality in the clinical setting. And also there could just be very big misinterpretations of the polygenic scores because they just don't have the same predictive utility. So what are we going to do about that? Well, there's huge efforts to address this, and I would say they're making good progress. There's still a long way to go. So one approach is to increase the ancestral diversity in our GWAS, so perform GWAS in other populations. Another very important thing is that we need to collect more diverse reference data. So I've mentioned that we're using summary statistics a lot in combination with this kind of LD reference data. At the moment, we've got LD reference data for some fairly broad populations, but it would be much more accurate if we had more diverse reference populations where we can model LD more accurately. And the third kind of line of attack to improve this situation is to develop methods that leverage information across different populations. So can we use these European GWAS in a better way to improve prediction in non-European populations? And yeah, there's a lot of research on that as well at the moment, and I will touch on that a little bit if I have time at the end. Okay, so that was a kind of very fast introduction to the world of polygenic scoring. It's got lots of applications. It's got quite complex methodology, and it's got some important limitations that still need to be overcome. And this is where GenoPred comes in. So this is just an animation showing this, but basically you take an individual's genetic information, you want to do some polygenic scoring to place them on this distribution of genetic risk. But this process of polygenic scoring is complicated, there's many steps, easily done wrong. The methodology is rapidly evolving. So it's quite challenging for people to get into to actually start using polygenic scores, but really shouldn't be that way. And this is why we developed GenoPred. So GenoPred is really a research program where we're trying to develop a framework for calculating clinically applicable polygenic scores with three kind of important sub-aims, I suppose: to benchmark existing methods, evaluate novel methods as they become available or we develop them in-house, and also importantly improve the interpretability of polygenic scores. So a little bit on GenoPred as a research program, and then I'll talk about the pipeline in a moment. We've conducted a range of studies looking at various different aspects of polygenic scoring methodology, trying to understand what's the best approach, how can we integrate different types of information to make them more accurate, how can we improve the interpretation of the polygenic scores. Populations, which is currently in progress. And I'll just flag this now that I've just submitted an abstract to the conference, the African Society of Human Genetics in Uganda next year, which I'm very excited to attend. So yeah, if you want to hear more about that, hopefully I'll be able to present it there. But if not, hopefully see you there and we can have a chat. So all of this work, I've throughout tried to make it as useful as possible for other people by making the code I've used publicly available on a website I've written. But there's many steps and it's often complicated, and really you're only going to be able to use that code if you've got some prior training and really it's not practical for a lot of people. So I then developed the pipeline version of GenoPred. So this is a software pipeline that's supposed to be easy to use. It implements leading methodology, and basically it allows people to go from very simple, widely available input data to polygenic scores in a robust and scalable way. I think there's a couple of things that I want to highlight here, which are of particular importance. One is that the polygenic scores are reference standardized. So this is really important because if you want to use a polygenic score in a clinical setting, and you've developed some sort of model that predicts risk based on polygenic scores, you need to be able to make sure you can calculate that polygenic score in the exact same way in a patient as you did when you trained your model. And this is one thing that this pipeline ensures by using reference genetic data throughout the polygenic scoring process, so you can reliably calculate the same score in the future. It's also very useful for research because it just avoids, it makes your research more reproducible. The other thing that I want to flag is that this pipeline is able to take GWAS data or target sample data from a range of ancestral populations, and it does this by using this reference genetic data in the default setting. It's a combination of the 1,000 genomes and the Human Genome Diversity Project, which has a fairly good kind of representation in it. There is a lot of improvement to be made on that, but it does mean already you can model data from a range of ancestries fairly well. So that's very useful. A little bit on how it works. It's a snake-make pipeline, which is very good for ensuring that your output is a result of the input. And if you change those inputs, it will tell you something's changed and it will recreate the output. So if you're involved in quite complex analyses, this is a super useful tool. It also uses Conda environments throughout, so it ensures your software environment is consistent and reproducible. It also means it's very easy to set up because it just installs all the dependencies it needs automatically. Everything is online and publicly available via GitHub. It's got version control, so if you use the pipeline, you will be able to reproduce your analysis down the road. If you want to do something extra that the pipeline doesn't do, you can also just make a copy of it and make the modifications if you're feeling up to it. So yeah, hopefully that will help people as well. And there's extensive documentation. So I've made a website which has lots of information on how to install it, use it, do whatever with it down the road. There's videos, there's technical details. So hopefully everyone has the information they need to actually use it and also write it up once they've used it in their publication. So I'm going to skip through some of these slides just for the sake of time, but they're just really trying to iterate that the kind of configuration of the pipeline is very simple. There's a few input files, and these input files have only a few parameters that are usually fairly easy to find the information for. It can accept data in a range of formats, so you can just give it what you've got and hopefully it will work. And of course, if it doesn't, I encourage people to contact me and I'll try and make it more flexible, but it already deals with the vast majority of file formats, so hopefully quite easy to use. I'm going to skip through this, but essentially you can run the whole pipeline in one line of code. So it's crunching a lot of steps, potentially hundreds of analyses into just one line of code. So very simple to use. I'm going to skip this point and this one as well. So it produces a range of outputs. These outputs can be found, the files will be saved, but you can also just read them straight into R. So again, trying to make it as easy as possible for people to use it, because a lot of people work with R. And yeah, I'll just show you one of these. So the pipeline at the end, this is kind of a bit of a fun thing, but also at the same time, it's quite useful for checking what the pipeline did. So it'll run like a range of analyses. Is this going to work? Right. Hopefully you can see my browser. Essentially, it creates these reports where it summarizes what the pipeline did. So it'll give you some QC information. So how many individuals were in the target sample, how many variants did they have that, and things like, there's just a few information. I'm not going to go through the details here. This is actually a report for just one individual. So it's kind of a specific report for one person, and it summarizes the probability that that individual belonged to the different reference populations. In this case, they were 100% likely to be European. Gives you some information about the polygenic scoring methods that we used, the GWASs that we used in these tables, and then it tries to summarize this individual's genetic risk based on the polygenic scores. And this is very important for interpretation. If you get a polygenic score, often it's quite an abstract number. This uses some previous research we did to put it on a kind of Z-score distribution, so showing the relative risk that individual carries. So this individual had a polygenic score lower than 95% of the general population, an ancestry matched population, importantly. And if you can actually convert this into absolute risk, what does that mean in reality for their risk of the disease? Well, actually, because the polygenic score explains so little variance, that individual still has the same probability of developing coronary artery disease as the general population. So really important to consider the variance explained by the score as well as the score itself. So that's just a demonstration of the reports. I'm going to give one example of using the pipeline, which was done by one of my colleagues, Michelle Camp. And I'm highlighting this one because I think it showcases the pipeline's ability to be applied to non-European populations, in this case, specifically African populations. So they used GWAS summary statistics from the Ugandan genome resource, so a kind of African ancestry population, and they calculated the polygenic scores into RWGen, another African ancestry cohort, and they were able to show, and they did all of this with GenoPred with relative ease. And the nice thing they showed with this in the title is that these polygenic scores were able to improve prediction of some cardiometabolic outcomes in African populations over and above the kind of non-genetic variables that might be used to predict. So it's basically saying that polygenic scores can improve prediction over non-genetic predictors of cardiometabolic outcomes. So quite a nice conclusion in a population that has historically been quite difficult to analyze with the tools available, but GenoPred makes this very easy. Oh, sorry, I didn't mean to do that. And then I've just flagged this as well. So what are the further developments? Well, one is to improve the include methods that can deal with GWAS from multiple populations. And another one is to incorporate methods that allow for functional information to be incorporated. So, for example, what if I want to calculate a polygenic score for people for schizophrenia, but only considering genetic variants that act through a specific biological pathway? And this is going to be, I think this is going to be a very important future direction, because it's going to allow us to kind of dissect heterogeneity within patient populations and potentially identify specific treatments for those individuals. So again, flagging, this is what I'm going to be presenting at the African Society of Human Genetics. This work looking at multiple populations and methods including multiple populations. And I think I'm going to leave that there. I'm going to skip. I was going to flash up some preliminary results, but I think for the sake of time and also suspense for the conference, I'm going to just jump through to my acknowledgements. I don't know how I can do this. Yes. So I'll end it there. I want to say a huge thank you to everyone that supported me throughout my research, and specifically the GenoPred. A lot of it was developed while I was working with Catherine Lewis and her team. I've got great feedback from several of the people within the team listed here. And also I want to thank the people that support my fellowship, which I'm carrying out at the moment, particularly Ammar Al-Chalabi, who's my primary sponsor. And also, of course, a huge thank you to King's College London, my institute, and the people that have funded this research, the Wellcome Trust, and also NIHR. So big thank you for listening. I'd be really happy to take any questions.
Thank you so much, Oliver, for that wonderful presentation. It was very well explained. Thank you, everybody, for listening in. The floor is now open to questions. If you have any comment or you want to ask a question, you can raise up your hand and I'll ask you to unmute, or you can equally write it in the chat box and then I'll read it out to Oliver. So to start with, we have the first question. Tumai is saying that Michelle Camp used PT plus C. Which is one of the methods the pipeline uses? So she's asking, is it possible to change the PRS calculation method in the pipeline for research purposes?
Yes, so absolutely. The pipeline implements currently, I think, 8 different methods. We decided in Michelle's study to use the p-value thresholding and clumping method because it's kind of the method that has the least assumptions. Some of these more modern Bayesian methods have a lot of kind of assumptions that we weren't sure would hold true in this African population that we were using. But the method can be swapped. You just tell it which methods you want. In this case, we just selected that one method, but you can also add in Mega PRS, SBASE-R, PRSCS, LDPRED2, all of the big hitters. And right now I can say that I'm looking at this in more detail using GWAS from different populations. And actually it seems like these more kind of fancy Bayesian methods do work pretty well in African populations as well. So, give it a go, but yeah, hold tight for more information on that, because I'm very close to, I would say, releasing some results showing exactly which methods work best in those different ancestral contexts.
Okay, thank you so much, Oliver. Ted Marsh, you can unmute and ask your question.
Yeah, thank you. Thank you very much. A very, very good presentation. My question to you is that it seems you have a gene-centric view. Rather, why don't you, or is there a way to incorporate lifestyle, social, environmental factor, clinical factors in the PRS? How much is PRS contributing to risk versus all other factors that we know have a bigger role than even genetics? So I was wondering how much effort you have added to put into including lifestyle or clinical or social factors, environmental factors into the model.
Yeah, great question. And you're absolutely right. genetics doesn't explain everything by any means, and you're going to be able to predict things a lot better using clinical variables most of the time. But there's, a lot of evidence showing that you can improve predictions over clinical variables alone by also adding in this extra variable capturing an individual's genetic risk for the outcome. And that's definitely my opinion on it. Like, I'm not saying it's going to it's not more important than clinical data at all. The polygenic scoring process doesn't take into account clinical information, but the way you model the polygenic score can be adapted to account for these non-clinical variables. So, for example, if I've calculated my polygenic score for coronary artery disease, I would then put that into a model which also has the other variables. So, you could take into account those clinical variables and the polygenic score in addition. The actual polygenic score calculation wouldn't change the way it is currently because we assume its effects are kind of additive and not interacting with other clinical variables, but there's no reason, that's the way the field is right now. But I think there's a scenario where you could find genetic effects that are specific to certain patient populations. I would say we're quite a long way away from dealing with that because typically our sample sizes are just about big enough to capture kind of main effects, let alone the interaction effects. But down the road, there may be scenarios where the polygenic scoring process actually does take into account other variables. But at the moment, it's just this kind of sum score, additive score. So yeah, it's definitely important to consider these clinical variables, but right now it comes at the end where you just kind of model those clinical variables alongside your polygenic score.
Okay, thank you so much, Oliver, for that. We do have a question from Malik. He's asking, what are the traits or the diseases that you tested with GenoPred?
So, good question. So, we've looked at a lot of stuff over the years. We initially looked at, I think, 11 common diseases in UK Biobank. And these were selected based on the availability of large GWAS, and they included things like depression, a few autoimmune diseases, height, BMI, breast cancer, prostate cancer, coronary artery disease. So those kinds of things. It's more recently, the study I'm doing right now, where we're looking across populations, we're using traits that have data in the different populations we're looking at, which are Europeans, East Asians, and Africans. So the traits we're using there are mainly anthropometric blood biomarker traits and kind of cardiometabolic traits, because those are the ones that are publicly available. And I would say, yeah, so we look across a range of traits. We try and look across traits that have different genetic architectures to see how well they work for highly polygenic or not very polygenic traits. But I would say the results are usually quite consistent in terms of which method performs best. They all, like, there's a certain group of methods that are very, very similar in how they perform, and they seem to be able to cope with a broad range of genetic architectures.
Okay, thank you so much for that, Oliver. Tomai is asking once again, by any chance, is your target population the AWI Gen study like Michelle did, or you had a different study target population?
So right now, I'm using the data I have available, which is actually UK Biobank. So I'm looking at predicting into European, East Asian, and African individuals within UK Biobank. So that's obviously got some limitations, but I think from a methods comparison perspective, it will still tell us a lot. I would love to test these models in other populations as well, like continental African populations or specific populations within Africa as well. So we'll see how the work develops. But yeah, RBGen would be a great opportunity for that.
All right, let's take some hands. Imane, you can unmute and ask your question.
Hi, thank you, Oliver. Can you hear me?
Yes.
Thank you. So my question is about mixed populations. In case we have an mixed population with individuals having different proportions, like different ancestral proportions. Some are closer genetically to reference groups, but others are highly admixed. Is it a good practice to generate risk scores, for example, using GenoPred, for those that are closer to reference populations presented by 1,000 Genomes and AGDP? Or like, what is a good practice in this case? And also, is there a way to generate these scores for these well-represented people, but then extrapolate to the others that are admixed, that are highly admixed, but within the same population?
Yeah, that's a really, really good question. And I would say another big kind of consideration when looking at genetics in many ways, but particularly polygenic scoring. So GenoPred currently doesn't handle that particularly well. It will try and assign an individual to the nearest population, there's a kind of threshold of how confident you are that person belongs to the reference population. So it's very much within the mindset of like, you belong to a population. For admixed individuals, they may be well enough captured for them to fit into that reference population, but it's very likely that their polygenic score will be less accurate than if it were kind of that admixture was modeled more appropriately. I've seen there's two kind of ways of dealing with it. Both of them are not currently implemented in GenoPred. One is to look at local ancestry, where you can kind of consider the effects of variants within a given region, and then look to see where that part of an individual's genome came from. So there's some really interesting work on that. It's not currently implemented in GenoPred, but if you're interested in that, look up Tractor, which is a very cool GWAS method that's trying to dig into that. I think polygenic scoring methods will follow suit at some point as the data becomes more available. And I also saw a preprint just yesterday, actually, that came out trying to deal with this admixture issue a bit in a different way, which could also be incorporated. But I would say this is still an issue for the field. There's not a good way of dealing with it currently.
Thank you.
All right, thank you. Thank you so much, Oliver. Well, Ted, you can unmute and ask your question.
Yeah, thank you again. You mentioned the pathway-based incorporating pathways. Are you looking at pathway for those SNPs with PRS in the model? How is the pathway incorporated to your score?
Yeah, good question. So this work is still in development, but the way people are doing it right now is they will annotate SNPs to genes and then calculate polygenic scores considering SNPs linked to genes within a given gene set or pathway. So for example, you could look at SNPs that are tagging genes that are within, the serotonin metabolism pathway or something like that. I think the sticking point there and the bit where it varies in the field and still yet to be determined what the best way is how do you link SNPs to genes? The approach that's currently implemented in software called Preset, which I think is the only software that does it so far, does it by location. So if the SNP is near a gene, it'll be identified as a SNP for that gene and therefore acting through the pathways that gene is involved in. Obviously, that's not ideal. There's probably better ways of capturing the functional consequence of a SNP. And we did look at this, and I think it captures one of the questions in the chat as well. We did a study on what are referred to as polytranscriptomic scores, where we considered the effect of SNPs or genetic variants on gene expression. And from that, that's a more kind of mechanistically informed way of saying what is the functional consequence of this genetic variant. And that allows you to calculate these things called polytranscriptomic scores, which are basically polygenic scores restricted to genetic effects mediated by altered expression. And I think they might be more, they might have some benefits if you then want to look at specific pathways as well, or other annotations, gene annotations. So yeah, this is early days, but yeah, if you're interested in looking at that, there's this software called Preset, and as far as I'm aware, they're the only people that do it so far.
Thank you so much, Dr. Oliver. So we're kindly requesting for some extra 5 minutes. We have some questions in the chat. If you have a question, you can wait around for Oliver to respond to you. So the other question that we have is someone is asking, what are polytranscriptomic scores?
Yeah, so I just touched on this, but it's essentially a polygenic score, which is restricted to genetic effects mediated by differential gene expression. So there's actually two types of polytranscriptomic scores. So there's the type where you impute gene expression in the target sample based on their genetic variation, and then you calculate kind of polygenic scores using that imputed gene expression. The other type is an observed polytranscriptomic score, where you've actually measured gene expression in that individual from their blood or some other tissue of interest. And then again, you calculate a polygenic score based on that observed gene expression. The kind of weights that you use for a gene come from a GWAS, a Transcriptome Wide Association study. So this is a way of predicting or inferring how differential gene expression will be associated with your GWAS outcome. So it's a little bit complicated, and I would say to get more insight on that, check out, there's 2 papers I wrote using both of those 2 approaches, and I'll put it in the chat. But yeah, that's, I think, a little bit, there's some nice figures which kind of illustrate it a bit more clearly. I think it's quite complicated to just say.
All right, thank you, Oliver. So we have a question from Azza. She's asking, how can we solve the population issue to use PRS in individualized medicine? She's saying, because, for example, in schizophrenia, the PRS is different between Africans and Europeans, is higher than that between European cases and controls.
Yeah, so I think I understand the question, and it's a good one. And I think the way to, there's 2 parts. One is you first of all need to interpret an individual's polygenic score in the context of an ancestry matched reference. So if you take a European individual, calculate their polygenic score, you can then standardize it according to an ancestry matched reference, aka a group of European individuals, and then say that individual's risk is 1 standard deviation higher than the average. And it's really important to do this using an ancestry matched reference, because genetic differences across ancestries can massively bias the polygenic score, not because they actually predict the outcome of interest, just because there's big differences in the allele frequencies between genetic variants across populations. So one is this kind of ancestry matched standardization. GenoPred does one version of that. There's a couple other ways of doing it, but that's something that GenoPred already does. The other thing you need to consider is that the variance explained by the score is going to be different in different populations. And this is much harder to deal with currently. But essentially, a schizophrenia polygenic score explains like 10% of the variance in Europeans, but probably a lot less in, say, an African population. So then when you interpret the polygenic score for a person, you need to interpret it in the context of an ancestry matched reference to say, where are they, relative to other people in that population? But then also, what is the change in absolute risk based on the variance explained by that score in that population? So it's a really tricky issue to deal with. And I think ultimately we'll need to do studies where we evaluate the performance of polygenic scores in the population that we intend to apply it to. So more research needed, basically. Currently, it's quite challenging, but clinical trials I think will be needed to really understand the true predictive power of a polygenic score in the relevant population.
Okay, thank you, Oliver. A quick one from Mustafa. How many ancestral populations is GenoPred being used for so far?
So currently it allows for 6 reference populations. They're quite broad populations, which is European, East Asian, South Asian, African, Middle Eastern, and what's called admixed American or Central American, basically. Some of those are more specific than others, and that's partly due to, it's due to the diversity within them. Europeans and East Asians are actually quite homogeneous populations, whereas admixed Americans, Middle Easterns, and particularly African populations are far more diverse because the label doesn't, it's a very broad label. But those are the 6 populations it currently captures. But I would say GenoPred, that's the default, but users can provide their own reference genetic data. So if they have data for a population they think isn't well captured by that reference data, they can use their own data to as the reference. So it's flexible.
Okay, so Vicky is also asking, what are the best practices for controlling false positives and overfitting in PRS calculations? And then she's asking, how does your tool address these?
Yeah, great question. So there's a few layers to this. It's really important that your GWAS summary statistics come from a population that's independent of your target sample. So if you do a GWAS in UK Biobank, you'll need to use a target sample that's independent to UK Biobank. Or you can do some sort of cross-validation where you split your data into training and testing subsets, and that will ensure there's no sample overlap there. So that's one side of it. But then there's also the important part of actually modeling the polygenic score. So a lot of polygenic scoring methods give you many polygenic scores derived using a range of kind of shrinkage parameters or p-value thresholds that the user then needs to deal with in some way to just get one score and one result. And again, you would do that using some kind of cross-validation framework. So you, in your target data, split them into training and testing, identify which polygenic score is the best in the training data, and then evaluate its performance in the testing data. And that will ensure you don't get an inflated variance explained by the polygenic score. Some polygenic scoring methods have this really useful feature referred to as pseudo-validation or the auto model, where it just gives you one score which has been kind of tuned using summary statistics only. And that avoids that problem of having multiple polygenic scores that you then need to model in some way. So for example, mega PRS has a pseudo validation score, so you just get one score. And actually, in our previous paper back in, I think, 2021, we show that usually these pseudo validation methods work pretty well. So if I was going to do a research project, I would just use those because it makes the analysis much simpler and it avoids the risk of overfitting really nicely.
Thank you so much, Oliver. Chris, you can unmute and ask your question.
Okay, all right. Thank you, Oliver. I just wanted to ask something a bit practical. So when I interfaced GenoPred, I realized it gives you, just like you showed, it gives you polygenic risk scores for each individual, like the R-squared value for each individual. But then how do we easily get the collective PRS, like such that it's easy to explain that maybe in this population, this R-squared is the collective one, and it's explaining this much variance for these people without so much manipulation? Because last time we were going through it and we had to like go to ARA and then get all the merge the files and all that. Is it something you're working on? That's one question. And then the other one is, I see you didn't include the C+T method, the one in Precise2, and I don't know, was it on purpose, or what do you have to say about that?
Yeah, so good question. So the first question, so the pipeline currently gives an estimate of what it thinks the variance explained by the polygenic score is, but it is very much an estimate. The method it uses to kind of guess the R-squared is not particularly accurate. So don't rely on that too heavily. Really, you need to, as I said before, you need to evaluate these polygenic scores using individual level data to see what the true variance explained is. So take it with a pinch of salt. There are new methods coming out which estimate the variance explained by the polygenic score, but they also come with limitations. So I would say basically don't ever take the variance explained, the estimated variance explained by a polygenic score to mean to be, you know, very accurate, unless it's been estimated in individual level data in the population you're interested in. That doesn't mean you can't use the polygenic scores, it just means don't rely too much on that variance explained estimate. And I think the second question was regarding why did we use the PT plus clump method in Michelle Camp's paper? And
No, why did you skip the C+T? The one that's in Precise2, yeah.
We kept it because it's useful. So it's a kind of assumption-free model. It's very robust to all sorts of issues in the GWAS because it's got this simplistic method where it just removes SNPs and LD and provides these p-value thresholds. It means there can be a lot of what's referred to as misspecification in the GWAS sum stats without running into problems. Whereas what we showed in our 2021 paper is that some methods, if you give them GWAS summary statistics that have had some sort of problem, like the SNPs, some of the SNPs are available for hundreds of thousands of people and some of them are only available for a few hundred. Those kinds of kind of things that can happen, particularly when you meta-analyze GWAS from many sources, it introduces all these kind of inaccuracies in the summary statistics. And this can lead some of the more advanced Bayesian methods to fail to converge, but it doesn't always tell you when it hasn't converged. So you get your score and actually it's not very accurate, but you wouldn't know that necessarily. So I usually tell people, use the best method, which right now I would argue is mega PRS or LDPRED2, but also use PT plus clump, because I think it's a very important sanity check that those more advanced methods are actually performing as they should. So we kept it in because it's got this really nice, robust quality, but yeah, it's not the most accurate.
Thank you.
Okay, thank you so much, Oliver. We have one last question from Mai. She's asking what are the potentials of using GenoPred in populations with high prevalence of consanguineous marriages and familial diseases. Seems like it's ready to use for these categories.
So, there's nothing stopping you using GenoPred for people in kind of consanguineous populations, but I think usually in those populations there's an increased prevalence of rare diseases caused by rare genetic variants that start to accumulate within these populations. Whereas polygenic scores use GWAS summary statistics, which typically only capture common genetic variation. So you can use a polygenic score in any population you want, but it's their ability to pick up familial diseases is going to be low. I think really you need to consider sequence data in those populations, otherwise you're not going to be capturing the variants that are relevant. So you could use it you can use them to some extent to help improve prediction or even predict the kind of penetrance of rare variants, but to get full information in that type of context, you'll also need sequence data to pick up the rare variants.
Thank you so much, Oliver, for that. That was the last comment that we had. Thank you so much for answering our questions so well and for the wonderful presentation. And I've shared the link for our evaluation, one evaluation form. For dear participants, you can feel free to fill the form, help us know how we can make this series much better for you. And on behalf of the organizers, I'd love to express our sincere gratitude to you, Oliver. Thank you so much for your time to share with us about GenoPred, and to the participants for attending this webinar series. We are looking forward to the next webinar, and above all, to meet Oliver at the African Society of Human Genetics come next year to give us more explanation physically now. So thank you all for attending today's webinar. Stay well and take care. See you. Bye-bye.
Thank you so much. And thank you to everyone for attending. Really good questions and engagement. Thank you so much.
Thank you too. Have a good time.